My name is Sue Wessler. I'm a professor of genetics at the University of California, Riverside, and my lab studies what is the subject of the talks today, and that are transposable elements. The title of the general lecture is A Dynamic Genome, and the title of this first presentation is Introduction to Transposable Elements. Here I will talk about the discovery of transposable elements and how this seemingly trivial discovery led to what is now recognized as a revolution in biology. So I start showing pictures that are integral to the talk. The first is a picture of Barbara McClintock, who is the discoverer of transposable elements, and I'll tell you more about her as the talk goes on. And the other is the corn kernels, which are also integral to the discovery. So I've divided the talk into three parts. The first is a description of the discovery of transposable elements by Barbara McClintock. The second is a little more detailed on how transposable elements actually move and increase their copy number in the genome. And the third is just how abundant transposable elements are in genomes. And this is really the part that that made transposable elements more than just a trivial discovery, and where it really has led to transposable elements being viewed as the major component of the genomes of higher organisms. So here's a picture of Barbara McClintock when she was a graduate student. And it's a picture of the lab of R.A. Emerson, which is who's the father of maize genetics. This picture was taken at Cornell University in 1929. And in the next talk, actually, we'll come back and revisit this particular area of Cornell. So here's a picture of McClintock. I think I'll show it here. And you could see that she wasn't wearing what women normally wore back then. That's a skirt. She wore knickers. I had the good fortune to know her later in her life, and I'll talk about that a little later. So the focus of their lab was corn and corn genetics. And one of the reasons it was such a wonderful organism to study genetics is it had an abundance of interesting visible characteristics, traits. And as we found out years later, one of the reasons for this diversity is that it has a remarkably diverse genome. So McClintock asked a very simple question, and this question is really at the heart of this talk and the next talk. And that is, why are these corn kernels spotted? Now, many of you may recognize this corn, although it doesn't look like the corn you eat, as the corn that you see during Thanksgiving. It's usually hanging up in supermarkets. It's Indian corn. It's known as Indian corn. And unlike the corn we eat, which is yellow, this corn is highly pigmented. But what's unusual about this particular corn kernel here, corn cob here, are the spotted corn kernels. And McClintock was interested in what is the genetic mechanism responsible for this phenotype, this spotted kernel phenotype. So here's a picture. A lot of McClintock's work, because she went on to be world famous, is available online, and you can actually access her notebooks. And this is a picture of her notebook with her typing at the bottom describing these kernels and the genetic basis for their unusual phenotypes. The one thing you'll notice is that the kernels are incredibly detailed. And that is one of the reasons that she was able to figure out so much about the behavior of the transposable elements that I'll talk to you about, because of the wonderful resolution of the phenotypes in the kernels. So I'm going to cut to the chase and tell you McClintock's solution to the spotted corn kernel question. And that is that she discovered that spotted corn kernels were caused by a new type of genetic element, a new mutation, and I've diagrammed it here. So if we start with the gene is mutant, but not due to a base pair change or a deletion. It's due to actually an insertion of a piece of DNA into the gene. And this insertion inactivates the gene. But unlike other mutations, such as I said before, base pair changes or deletions, this mutation is reversible. So and the way it's reversible is that the piece of DNA, as we call it, the TE or transposable element, and you'll see this abbreviation throughout TE, The TE can insert into the gene, and it can then excise from the gene. When it inserts, we have a mutant phenotype. When it excises, we revert to a wild-type phenotype or normal phenotype. What I've drawn here is a spotted corn kernel. And I've tried to explain how the different sectors in the kernel arise. So what you see at the top is a mutant gene. That is, and so we can imagine that this gene is a gene responsible for pigment biosynthesis. So that if the gene is mutant, there's no pigment. The kernel is yellow. If the gene is wild type, normal, the kernel is purple. So to explain a spotted kernel, we have to have a reversible phenotype, a reversible mechanism. So what you see is that the colorless areas are due to the gene at the top. That is, cells that have the gene at the top. That is a gene that is disrupted by a transposable element, so it cannot produce the products necessary for pigment biosynthesis. And so we end up with the yellow, unpigmented areas. However, during kernel development, the transposable element can excise from the gene. And when it does that, it restores expression of the gene, and we end up with the spotted kernels. And so because kernel development is very regular, that is that unlike in animal systems, plant cells, when they divide, they don't move around. They divide and they're literally cemented in place. We end up with these sectors. So a large sector is due to a cell in which the transposable element has excised from the gene, and all of the mitotic progeny then can express the gene. So we end up with a sector much like a clone on a on a petri dish of bacteria. And as I said before, the rest of the kernel are from cells that still have the transposable element in the gene. So McClintock was able to use the behavior of transposable elements and the detailed resolution that the corn kernels facilitated to understand something about the behavior of the elements, and that's shown in this slide. So what we have here at the top is a gene that's pigmented, and it's pigmented because the color gene is wild type. That's one allele, a wild type gene. We have another allele where we have what I call an NTE. It stands for nonautonomous TE, and I'll explain that in a second. What you're going to see here is there's two different types of transposable elements. One is autonomous, one is non-autonomous, and these slides will hopefully clarify what I mean by that. So in this case, we have a transposable element sitting in a gene, but the transposable element, which is non-autonomous, is not able to move on its own. However, that transposable element can move if there's a second transposable element in the genome, and that's shown here as a TE, and this is elsewhere in the genome. It could be on a different chromosome. It is not near this color gene. But that transposable element activates the non-autonomous element, and it causes it to excise from the gene, and we end up with a spotted kernel. In the absence of that autonomous element, as you see above it, the colorless kernel, the transposable element cannot move. Okay? That's why we call it, it's non-autonomous. And finally, the last situation is we can have autonomous elements inserting into genes. And that's shown here. We have the autonomous element inserting into a gene, and because the autonomous element makes everything that is needed for transposition, that kernel will be spotted. To review, McClintock not only discovered transposable elements, but she also discovered that there are different types of transposable elements. And in this case, we have the autonomous element, and that's defined as an element that provides everything needed for transposition, and non-autonomous elements, which can only move or transpose in the presence of the autonomous element. That is, if the autonomous element is in the genome simultaneously. So McClintock made her discoveries initially in the 1940s, very long time ago. But it took a long time for the scientific world to catch up with her. That's why I say here she was well ahead of her time. So in the 40 or 50 years after their discovery, transposable elements, which were initially only recognized to be present in maize, were found in many organisms, in fact, in virtually all eukaryotes. In the 1950s, transposable elements were discovered in Drosophila, fruit fly. In the 1960s, they were discovered in bacteria, in E. coli. And in the 1970s, they were discovered in the human genome as a cause of some mutations in the human genome. And we'll talk a lot more about that later. So here's one of my favorite pictures. It's a picture of transposable elements in action in a rose in the Napa Valley. And another picture from my colleague, Tom Garatz, is a picture of a petunia flower. And the reason I show you this is that if you look in gardens, rose gardens or other gardens, you will notice these phenotypes. They're not just patterns, as you see with a lot of flowers. They're actually sectors. And in case these sectors are completely analogous to the spots on the corn kernels that I showed you before. So McClintock, as I said, it took 40 years really for the world, the scientific world, to recognize that her discovery in corn, in maize, was true for most eukaryotes. And in fact, what McClintock discovered was that there was more in the genome than just genes. She discovered a new component of the genome. And I've drawn that here as a chromosome before McClintock, showing these rectangular boxes, and those are representations of genes. So before McClintock, people thought, when they thought about it at all, that the genome, that chromosomes were essentially genes, sort of lined up like beads on a string. After McClintock, it was recognized that there was another component in the genome. And that component, as I draw on as black ellipses here, were the transposable elements. So McClintock recognized that these transposable elements that were moving around were not coming from the environment. They're not viruses that are infecting the organism. They actually are residents of the genome, and these we now know are residents of most eukaryotic genomes. So for this discovery, McClintock was awarded the Nobel Prize in Medicine or Physiology in 1983. Now, Nobel Prizes are frequently awarded many years after the discovery. 40 years is a very long time, even for Nobel Prizes. And this really is for the reason that I said before. She was really ahead of her time. The other thing is that Nobel Prizes are frequently awarded for up to 3 people. If she was awarded the Nobel Prize by herself. And this really recognizes that this was her discovery. So here's a picture of her. She lived from 1902 to 1992. I had the wonderful fortune of knowing her for the last 10 years of her life and continuing on with some of her discoveries, and I'll be talking about that in the next talk. She also commented on how her life really encompassed the history of genetics. So Mendel's laws were rediscovered in 1902, early part of the 1900s, which was the year of her birth. And she lived long enough, first of all, to be recognized for her great discoveries, but second of all, to actually see the world enter the genomics age, so the age of DNA sequencing. And that's what we'll talk about in a minute. So what I'm going to do now is tell you a little about how transposable elements move. So we're going to move from the genetics to the molecular biology. So here is a diagram of what a generic transposable element looks like. And so they're very simple genetic systems. So this is a piece of DNA. It varies from a couple of hundred nucleotides to several thousand base pairs. Transposable elements, this is an autonomous element. So remember, this is the element that can move on its own. It encodes everything needed to move itself and to move a non-autonomous element. So when it says everything needed, that is a single protein it encodes, and that's called transposase. And I'll tell you in a minute a little bit more about what transposase does. The element is flanked by special sequences, which are called terminal inverted repeats. These are sequences that are the same sequence forward and then flipped over backwards. And I'll show you in a minute what the functional significance of that is. And the whole element, in fact, is flanked by a target site duplication, a TSD, and I'll show you in a minute how that is derived. So I mentioned that there are autonomous elements and non-autonomous elements. non-autonomous elements, which cannot move on its own. They cannot move on their own. And they require an autonomous element to provide the stuff needed to move them. And you can see here that what we have as a non-autonomous element is sometimes, but not always, a defective version of an autonomous element. So what I've drawn here is a deletion that, that has been sustained, which prevents the non-autonomous element from making transposase. The transposase that's made by the autonomous element, as we'll see in a second, can influence both the movement of the autonomous element and the non-autonomous element. So that's shown in this slide. So what I've shown here is the transposase, the autonomous element, which is at the top, encodes a single protein, and that protein is a transposase. And what the transposase does initially is it binds to the ends, the terminal inverted repeats of both the autonomous element and the non-autonomous element. So one of the functions that the transposase has is as a DNA binding protein. Okay, so this cartoon sort of will take you through the steps in the transposition of a DNA, of a transposable element. So I've shown here that this is what we saw in the previous slide, but in a more abbreviated form. We have the transposase proteins bound to the end of a transposable element. Those transposase molecules come together and form a dimer. They then cleave the transposable element out of what's called the donor DNA, out of the rest of the DNA. And that entire complex, as you see here, then can insert somewhere else. So what we end up with is a transposable element in a new location in the genome. So I want to define another term, and that is a transposable element family. And this we'll see a lot more in the next talk. So a transposable element family, as you saw before, has autonomous elements and non-autonomous elements. And there can be lots and lots of members of the family. So we can have one or more autonomous elements, one or more, many non-autonomous elements in the genome. And as you see here, the structure of the non-autonomous element can vary. Sometimes it'll be a simple deletion of the transposase region. Sometimes it could be more extensive. And sometimes there may be none of the only thing it may share with the autonomous element are the terminal inverted repeats and the length of the target site duplication. So what I'm going to do in this slide is to show you how the target site duplication arises. So what you see at the top is going to be a new insertion site of a transposable element. So this is a piece of DNA where the transposase is going to bind. Now, we talked before about the transposase binding to the ends of the transposon. Now, this is different than that. This is the target site. This is where the transposable element is going to insert. The transposase has a lot of different functions built into this single protein. One of the functions is to cleave the target site. And it cleaves it in a way much like restriction endonucleases. It makes a staggered cut. So you see that here. It cuts on the two strands of the DNA at essentially the same sequence. And when those strands come apart, you see that we're left with these overhangs, these sequence overhangs. It's into this region that the transposable element inserts. Then we're left with these gaps on the side. Those gaps are filled in by host enzymes. And because of this reaction, the transposable element, which is shown in green, is then flanked by a repeat, a target site duplication, which is the length of the duplication. In this case, we show that the transposable element is cleaving 5 base pairs. It's a staggered cut of 5 base pairs. The repeat sequence will be 5 base pairs. Different transposases have characteristic staggered cuts. So some transposases will cut 3 base pairs apart, some will cut 8 base pairs apart, and the resultant transposable element will then have a target site duplication that is the length of the staggered cut. So again, here to remind you, here's our transposable element family. And this is to show you that the families like all families, like human families, can be different, can look different. What we see here is there could be multiple autonomous elements in the genome. There could be many, many, many non-autonomous elements in the genome. And we'll see more in the second talk. The other thing that differs is that genomes can and do have multiple families of transposable elements. So I've shown you here at the top the family we've been working with, where the transposase, the blue family, where the transposase is produced and binds to all the yellow terminal inverted repeats. At the same time, the genome could also have a different family. And what I've shown the green family at the bottom, the transposase from this family binds specifically to the purple terminal inverted repeats. So in this way, there could be multiple families that coexist in a genome that have really nothing to do with each other. What I want to do now is, in a few slides, take you through what I haven't explained to you. I've told you that there are multiple copies of transposable elements in the genome, but I haven't explained how a transposable element can increase its copy number. Because in fact, what I've told you is sort of the opposite. I've told you that a transposable element at one place excises and moves someplace else. And I'm not really great at math, but I can figure out that that won't increase the copy number. You start with one, you move from one site, you move to another. So I want to show you in the next couple of slides is just very simply, I'll show you a schematic on how, by doing that, a transposable element can actually increase its copy number. Because the copy numbers of transposable elements will be a major part of the talk, the rest of this talk, and the talk that follows. So what I've drawn here is, at the top, I have a transposable element at a particular site in the genome. That region is now, the DNA is being replicated. And what we see is the familiar replication fork. So here are the sister chromatids. Okay? So what's happened, and this is the same thing at the top, we're replicating. The transposable element is going to move from one of the sister chromatids after replication to another site, to the other sister chromatid. And that's shown at the bottom here. Okay? So I've redrawn that at the top. And what happens after this is kind of neat. The site, the empty site here, sometimes it remains empty. Sometimes the host will use the transposable element on the sister chromatid to copy it into that empty site. And that's what you see at the bottom here. So when these chromosomes, when replication is finished and we end up with two double-stranded daughter strands, what we have is the top strand has one transposable element and the bottom strand has two transposable elements. So we've gone from a situation where we've had one transposable element to one that now we have two. These chromatids will separate and they will go into separate cells. So one cell will have two transposons, one will have one. And I think in the next slide what I've done is I've summarized all of the steps. So we start out with a single transposable element. There's replication, there's transposition that occurs from one sister chromatid to the other. There's repair of the empty site using the transposon from the sister chromatid, and there's separation of the chromatids, and we end up with an increase in one transposon in one of the cells. That's one mechanism. There's another mechanism which I've just drawn as a shortcut here, and that is, you see at the top, just where we started before, we have a transposable element, we have replication. We then have transposition not from a replicated site into another replicated site, but instead from a replicated site into an unreplicated site ahead of the replication fork. What happens then is, again, we'll have the completion of replication, and we end up with the separation of strands, and we end up again with the gain in the number of transposable elements. Okay. So I have told you so far, I've focused on what's now known to be one of two classes of transposable elements that are in the genome. The elements that were discovered by McClintock and that are responsible for the unstable, the spotted kernels, the sectored flowers, those are caused by the element type that I've shown you here with a transposase with terminal inverted repeats. But there, in fact, is another class of transposable elements that I'm not really focusing on in this talk, and I'm only going to mention it briefly. But these are, in fact, incredibly abundant transposable elements. They're called they're called retrotransposons. And these are called now class 1 transposons, whereas the DNA transposons that McClintock studied are called class 2 transposons. Now, the retrotransposon is characterized by terminal inverted repeats, long terminal inverted repeats. So the ends of the element are not inverted repeats like the DNA transposon, they're direct repeats, which are shown here. They also have a target site duplication because the insertion occurs in the same way as the insertion of DNA transposons. And as you see, I'm calling them DNA transposons and RNA transposons. Now the reason is they're named for the intermediate in transposition. So a DNA transposon excises from one site as a DNA element and moves elsewhere. In contrast, an RNA transposon or retrotransposon, the intermediate is RNA. And I've summarized that in the next slide. Okay. So here I've shown you a retrotransposon inserted into a chromosome somewhere. The element is transcribed much like a gene. That RNA then is converted into, it's copied into DNA, into a DNA copy. And this is copied by an enzyme called reverse transcriptase, and it's encoded by the retrotransposon. That is then converted into a double-stranded DNA molecule, and that double-stranded DNA molecule inserts elsewhere in the genome. So this is a lot easier than the DNA transposition mechanism I showed you before. In essence, you could think of a retrotransposon as like a printing press. It makes RNAs, and each of those RNAs potentially could be converted into a double-stranded DNA, and that double-stranded DNA can insert elsewhere in the genome. So a single element, because there can be many, many transcripts that come from a single element could potentially lead to hundreds and hundreds of new integrations, of new copies in the genome. Okay. So I've told you about the discovery of transposable elements. I've told you about something about how elements move and increase their copy number. Now I want to sort of wow you with something that I think has been one of the major findings of this era of genomics, and that is just how many transposable elements there are in the genomes of higher organisms. So when we started sequencing genomes, and when I say we, I'm using the general we of the scientific community, it turns out that the largest component of genomes are derived from transposable elements. 50% of the genomes, of the human genome, of the chimp genome, of the mouse genome, more or less 50% are derived from transposable element sequences. Plants, especially flowering plants, have even higher proportions of their genomes as transposable elements. The maize genome, over 75% of the genome is derived from transposable elements. The barley genome, and we'll be talking more about these creatures in the next talk, the barley genome is 85% transposable elements. And even more remarkable, the iris genome is 98% transposable elements. And I don't know about you, but if I look at an iris plant, I mean, they're beautiful. There's nothing that would tell us that their genomes are just largely transposable elements. I want to give you a feel for how many transposable elements there are in the human genome. So our genome is comprised of 2.5 billion base pairs. Let's call them A, G, C, or T. Let's say that's 2.5 billion letters, like A, B, C, and D. Okay. So if they're letters, let's start filling up some books. So this is equivalent to about 1,000 textbooks of 1,000 pages each. No pictures, all right? Only 20 to 40 of those 1,000 textbooks contain all of the genes necessary to encode the proteins that make us up. 500 of the 1,000 textbooks contain sequences that are derived from transposable elements. So it's pretty stunning. To give you an idea of just how many transposable elements there are and where they are, I'm showing you an example of a typical human gene. And what you see are the green boxes, which are the exons, the coding regions, and the blue areas, which are the introns. Let's look and see where there are transposable elements in this gene, because there are transposable elements in 70 to 80% of our genes contain transposable elements. So this shows you all the different places that this gene has transposable elements. What you'll notice, it's kind of hard to tell in this slide, but that mostly the non-coding regions, the non-exonic regions, contain some transposable elements. In fact, some human genes have over 100 transposable elements in their introns. So transposable elements are really everywhere. So what I want to show you in the next slide is how transposable elements can diversify a group of very, very closely related organisms. And for this, I'm going to go back to plants, and I'm going to show you a group of organisms that we're very familiar with, the cereal grasses. You may not know, I came from New York City, so I didn't realize that all of these were in fact members of the grass clade. They are rice, sorghum, maize, and barley are some of the most important organisms on this planet for human calories. And what you see here, we've talked about maize a lot. The maize genome size is 2,500 megabases. That's about the same size as a human genome. However, what you see is that very relatively closely related organisms, the rice genome has a genome size that's almost 10 times smaller, 350 megabases. Sorghum is twice the size of that, 700 megabases. Barley is 5,000 megabases. These genomes have gone through a remarkable explosion. And what's responsible for that largely is the increase in the genome size due to transposable elements. How do organisms function with that much stuff? There's actually three major reasons for the success of organisms despite being crowded with that many transposable elements. The first thing is that most of the transposable elements in the genome are dead. And when I say dead, I mean they can't move. They don't move anymore. They probably haven't moved for a long time. And they're dead because they're mutated. They contain mutations. So every single generation, mutations are introduced into our DNA. Mutations that occur in genes will lead to problems for the organism, and they're selected against. However, mutations that occur in transposable elements just accumulate. It's not a problem to the organism at all. So we end up, most of the transposons, the vast majority of the 50% of our genome that's transposable elements, are not able to move around anymore and will never move around. They will just sort of, as we say, senesce. The sequences will mutate and mutate until there's really no trace that they ever were derived from a transposable element. Okay. The second way that organisms survive, and it's really how transposable elements survive, is that transposable elements have evolved mechanisms that allow them to insert into places in the genome that won't harm the organism. So for example, one safe haven might be into another transposable element. So if a transposable element inserts there, it's not it's not causing a mutation in any of the genes necessary for the organism to survive. And we'll talk a lot more about that in the second talk. The final thing, which I'm just going to allude to briefly here, is that the host has a way to fight transposable elements, and it's a very sophisticated way. And the host silences transposable elements using epigenetic mechanisms and inactivates them. And I'll show you on the next slide. So this is just an example of a region of the barley chromosome. And what you see are the few, remember I said that 85% of the barley genome is derived from transposable elements. And this is how a region of the genome might look. So here we have a few genes, the little blue boxes over there. And what you see stacked up here are all the transposable elements. They're the vast majority of this particular region of the gene. So, but if you look at, this is a flat version of the DNA. If you actually look at the three-dimensional structure, at the chromatin, the way the genome exists, what you find is that the region where these transposable elements are clustered, in fact, is a tightly compacted region of the genome. It's what's called heterochromatin. There's very little, nothing really is, this is a host response. It condenses the chromatin and prevents the transposons from making the transcripts and the proteins needed for it to move. So they're said to be silenced. This in addition to the fact that these transposable elements are accumulating mutations. In contrast, the region where the host needs the genes to be expressed, those are euchromatic regions. They are less condensed. So the genome, a chromosome, is composed of regions that are very, very densely compacted, and that's generally where the transposable elements are, and regions that are much less compacted so that the host can access those to make the gene products needed for its development in life. So like many things with transposable elements, McClintock was ahead of her time. She didn't only discover transposable elements, but she proposed that they had a role in generating diversity. And this scenario had the following components. That transposable elements in the genome usually do not move around, because if they did move around, they would cause mutations. That stress conditions may activate transposable elements. Now, when I say stress conditions, I'm thinking more, I'm not talking about like driving in rush hour traffic. I'm talking more about climate change. That might be one thing. This is a scenario. This is not proven by any means. This was her ideas 20 and 30 years ago. So genomes have a stash of transposable elements. Most are inactive, some are active. And the host is keeping them inactive by these epigenetic mechanisms. But if something happens to the host, some of these, or the host population, some of these transposable elements can be activated to move around. The significance of that is that the movement of transposable elements will generate genetic diversity. And it'll do this by increasing the frequency of mutation. So what we end up with is a population that is now more diverse because of the movement of transposable elements. There's new mutations in that population, and it's possible that some of those new mutations may be adaptive, may help the population survive this dramatic change in climate or whatever. So I want to end by essentially saying to you that the way I think of transposable elements is that they shake up the genome. And the genome is inherently conservative. So they shake up an otherwise conservative genome in ways that we are just beginning to understand and in ways that I will go into in the next talk.